Metabolic stress and toxic load. Just as an internal combustion engine operates as an input-output system, the human body functions in a similar manner. The fuels we consume, glucose, amino acids, and lipids, fats, enter the bloodstream through digestion and are transported to the liver, lymphatic system, and kidneys for processing. As these nutrients circulate throughout the body, they undergo catabolism, the biochemical breakdown of molecules to release energy. In the case of glucose, this process begins with glycolysis, the progressive conversion of glucose into pyruvate. Under conditions of metabolic stress, pyruvate is preferentially directed toward lactate production, which serves as a metabolic marker of nutritional or energetic stress. Proteins are processed by pancreatic enzymes that degrade them into peptides and subsequently into individual amino acids. Alterations in citrulline, ornithine, and arginine can provide insights into hepatic detoxification capacity and the efficiency of the urea cycle. Lipids, primarily in the form of triglycerides, may be utilized as an energy source through V-oxidation or incorporated into the structural architecture of cellular membranes, phosphatidylcholines, phosphatidylethanolamines, ceramides, among others. When lipid processing becomes inefficient, it may contribute to metabolic overload and a pro-inflammatory tendency. The body must continuously eliminate excess nutrients and metabolic byproducts. Under balanced conditions, characterized by adequate caloric intake, sufficient micronutrients, and appropriate rest, the organism maintains homeostasis, internal stability, through highly efficient biochemical pathways. However, during periods of metabolic stress, such as malnutrition, micronutrient deficiencies, overnutrition, infections, intense physical activity, or chronic inflammation, these elimination systems can become overwhelmed. As metabolic toxins accumulate, overall metabolic efficiency declines, creating a cycle of escalating stress and diminished physiological resilience. This vulnerability is more pronounced in individuals with inborn-like errors of metabolism, in which subtle enzymatic inefficiencies reduce the biochemical safety networks that normally help the organism cope with stress. These enzymatic redundancies function much like a seatbelt or an airbag, rarely noticed until the system is challenged. Through the use of targeted mass spectrometry, it is possible to measure key metabolites in the blood to assess how effectively the organism responds to metabolic stress. Patterns observed in acylcarnitines, the glutamine-glutamate ratio, and biogenic amines provide valuable insights into the efficiency of nutrient processing and the clearance of potentially toxic metabolic intermediates. Markers of metabolic stress may be associated with fatigue, reduced exercise tolerance, inadequate recovery, cognitive brain fog, increased inflammatory tone, or heightened sensitivity to dietary changes. Early identification of these metabolic patterns can help guide nutritional strategies, micronutrient support, and lifestyle adjustments aimed at improving detoxification capacity and restoring metabolic efficiency.